Hello, ladies and gentlemen. This is Dan Montano. I'm Chief Executive Officer of Zhittya Genesis Medicine. Today, we're having a webinar on peripheral artery disease, the utilization of therapeutic angiogenesis to treat this vascular disorder. Dr. Jack Jacobs will be giving the medical and clinical presentation in a moment. What I would like to do is a quick overview. I'd like to welcome and thank all of you that are here. I know that some of you are in Europe, so it's a lot later than it should be, but we thank you for your attendance. Peripheral artery disease is the third leading cause of cardiovascular disease. And for those of you who have ever known anybody that has it, it's a very painful and uncomfortable situation. We've been working on this for about 20 years, and Jack is going to give you a presentation. At this point, I'd like to introduce Dr. Jack Jacobs. Jack has been my partner for over 20 years. Jack is going to be, that's Jack there. Jack, come back on. That's Jack there. And he's going to be giving the presentation on peripheral artery disease. Jack, please take over.
Okay, thank you, Dan. I'm going to be talking today about using therapeutic angiogenesis to treat peripheral artery disease, a very painful disease of our legs. Okay, we all have about 60,000 miles of blood vessels in our bodies, which is enough to wrap around the world 3 times. And obviously, if we have blockages in those arteries to our legs, we're going to get what I'll be talking about today, peripheral artery disease. A few weeks ago, I talked about blockages to the heart, resulting in coronary artery disease. And actually, in a few weeks, I'll be talking about some newer evidence that blockages in the brain can lead to such things as Parkinson's disease, Alzheimer's, and stroke. Now, we're broadcasting from Las Vegas, so every once in a while I might have to take a sip of water. It's dry and somewhat hot here. Okay, so in fact, there are 75 human diseases that result from a lack of blood perfusion to a tissue or an organ. Some of those are obvious, as I mentioned, coronary artery disease from blockages in the coronary arteries. Blockages in the carotid arteries can lead to stroke. Newer evidence indicates Parkinson's and Alzheimer's. And one way to increase blood perfusion in those diseases is to grow new blood vessels. And that's a process called angiogenesis. It's a natural process. We use it all the times in our bodies. Let me just give you an example. The skateboarder fell off his skateboard. He scraped his elbow there, and he's going to use natural angiogenesis to heal that. So let's take a little bit closer look at that. Let's say this is the cut on his elbow. This, let's say, is a scab. We look underneath that scab, and here we see angiogenesis at work. All these nice new blood vessels here. These are formed as a result of the drug that I'll be talking about in a minute. FGF1 stimulates the production of all these blood vessels, and this is going to heal his wound, growing new skin cells, new nerves, new blood vessels, sweat glands, hair follicles, whatever you need to regenerate skin as an organ. And probably the most potent stimulator of this process is FGF1. Okay, this is the drug we're developing. It's a biological, it's a protein. I actually worked on purifying this from calberine a number of years ago when I was a graduate student. So let's see how this promotes angiogenesis. Okay, here is a new blood vessel, or here is an existing blood vessel. Here's our FGF1. All these molecules of FGF1 are coming and binding to the FGF1 receptor right here. And that starts an explosion of metabolic activities in that blood vessel. We start getting endothelial cell division here, and lo and behold, here's a new blood vessel beginning to form. It's a tube, and then it elongates to form a new blood vessel. So this is how we get better perfusion into damaged tissues, by growing new blood vessels. And this process can be repeated thousands of times along this vessel. So let's look a little bit more at this interaction of FGF1 with its receptor. So here's FGF1, kind of like a key going into a lock right here. And once that occurs, we get all these processes activated within the cell. And this all leads to cell division and the production of those tubes and new blood vessels. So I'm often asked when we give this drug to people, why don't we get blood vessels formed throughout the body? And that's a very important question. We don't want blood vessels growing in our eyes, in the retina of our eyes, or clogging up our kidneys. So let me explain a very important concept of how our drug works. And this is what is behind the safety of our drug. Let's think about developing a disease, let's say heart disease, for example. So our hearts go from being healthy to having the coronary arteries blocked in the disease, and that occurs over time. So over time, the heart becomes ischemic or starved. It's starving for oxygen, more blood perfusion. So the heart is crying out for more blood vessels. And what happens there, this is a healthy heart tissue. Here's the FGF1 and its receptor, not much going on there. But over time, as we develop, or someone develops heart disease, you can see we end up in this situation where the FGF1 receptors have been tremendously upregulated. Researchers have shown in this condition, the disease condition, there are about 10,000 of these guys per cell versus only about 100 here. So what the heart is doing, it's asking, it doesn't need new blood vessels here, but here it does. It's crying out for blood vessels, but there's just not enough FGF1 in the system to get that explosion of metabolic activity. So this is where we introduce FGF1 as a drug and what's called therapeutic angiogenesis. So here's the artist's rendition. Here's the cells crying out with their FGF1 receptors upregulated. Here we come in with FGF1 for therapeutic angiogenesis, stimulating new blood vessel growth. Now, I've given a number of webinars on this. We've used therapeutic angiogenesis to treat diabetic foot ulcers. Today, I'll talk about PAD. A few weeks ago, I talked about coronary artery disease, and I just want to show you a short video on that, because these two are very, very much related. Here, we're going to be injecting the drug into the leg muscles. In this indication, we injected the FGF1 directly into the heart muscle. And so we have nice data, clinical data on this. And this is a trial that I supervised a number of years ago, so I'm very familiar with the details. We injected FGF1 directly into heart muscle, and we grew new blood vessels in the hearts of patients. Let me show you an ABC News clip from one of our clinical trial sites in Cincinnati, University of Cincinnati. ABC News went there and interviewed some of the patients and also our cardiologist, Dr. Lynn Wagner.
But since receiving an experimental treatment for his blocked arteries, his pain is gone.
I really feel great.
Duke was one of the first heart patients in the country to be treated with a protein actually capable of growing brand new arteries. The genetically engineered protein is injected directly into the heart. Within days, a network of new vessels begins to grow around the blockage, increasing the blood supply. Dr. Lynn Wagner showed us the changes in one patient's heart.
We see a small, narrow main artery and not very many secondary and tertiary arteries. This is after the treatment. What we're now seeing is new blood vessels growing here off the end of this artery.
And the patients themselves?
Symptomatically, they're improved within a couple of weeks of the treatment.
Just ask Constance Donnelly.
Oh, I feel wonderful. I've never felt so good in the last 5 years.
But doctors already see potential in other cases where the blood supply needs a boost, such as strokes and diabetes.
Okay, that woman we saw there in the video, she was actually in the lowest dosing group. We had 3 dosing groups, and low, medium, and high. She was in a group that we thought got about 1/10 of the optimal dose, and she really responded well. She came in as a cardiac cripple, so her receptors must have been tremendously upregulated. So let's go on and talk about peripheral artery disease. This is a disease, just like in the heart with coronary arteries, we get blockages in our leg arteries. Approximately 8.5 million people have this disease in the United States over the age of 40. Actually, in the worldwide, there's over 200 million cases of PAD. and 2/3 of those are occurring in lower income or middle income countries. So it's a real global problem. As you can see in this chart here, PAD increases as we get older, both in men and women. And look there, by the time we're over 80, large numbers of Americans have this disease. Okay, if you have PAD, it's a sign of serious vascular disease in the rest of your body. Look down there at the bottom right. 60% of people with PAD also have disease in their heart and in their brains, cerebral vascular disease, where if you have heart disease, only 24% of those patients have disease in their other vascular beds. So once you have PAD, it means your vascular system is in serious shape. Let's look at some of these risk factors for PAD. A great one is a history of cardiovascular disease. If we go down that list, we see high cholesterol, smoking, either past or current, is a very high risk factor for developing PAD, as well as diabetes. We know diabetics often develop PAD, as well as hypertension. Race and PAD, it's known that African Americans, in red there, have a much higher risk for PAD, over 30% of them as they get into their 80s and 90s. This most likely is due to genetics and diet. Okay, what are the treatment options that we have today for PAD? In the United States, probably the most popular option is surgery. Two types of surgery. They can actually go in and remove the clot from the leg, or they can do a bypass. Just like in the heart where they bypass a coronary artery, they can do a bypass procedure in the leg artery, and usually they will harvest veins to do that, but even if they can't harvest veins, or synthetic veins, synthetic grafts they're called, which can be used to treat PAD. In yellow there, same as in the heart, you can put a stent, a balloon, catheter, kind of balloon inflates, gets rid of the clot, and then we put a metal stent in to keep that vessel open. Drawbacks for both of these types of surgery is what's called restenosis, redeveloping of a clot in the artery. And so I've seen papers where after 5 years, that reclotting can happen in 40 to 50% of patients. Those who can't be treated this way are managed just medically with blood thinners and anticoagulants. Okay, so here's the main symptom of peripheral artery disease. It's called claudication up top there, leg pain on exertion. Greater than 50% of people with PAD will have leg pain. Some will have atypical claudication, but most will have typical. Typical claudication there is, say you walk up a flight of steps, you develop leg pain in your calf muscles. And that will typically last for 2 to 3 minutes. And then if you rest, it'll go away in 10 to 15 minutes. So that's what's called typical claudication, the most common symptom of PAD. Now in red there, we can see 5% of people with PAD go on to develop what's called critical leg ischemia. And that's a very serious form of PAD. You're at risk for gangrene in the leg, lower limb amputation. So this is probably the most serious form of PAD. So let's look at the spectrum of symptoms with PAD. Starting on the left, they're normal, healthy muscle, no problems running or walking up steps. We then, in the beginning stages of PAD, will develop fatigue and heaviness in our calf muscles as we, say, walk for longer than a few blocks. Then claudication comes in. That's leg pain on exertion, and that could be mild, moderate, or severe. And it is the claudication people will probably be treating in our first clinical trial, looking at PAD. And then as the disease progresses, you get into the critical limb-threatening ischemia there, where you have pain in your legs all the time, even lying in bed. You develop wounds, almost like in the diabetic foot ulcer, you develop ulcers that do not heal due to a lack of blood perfusion. And then finally, impending or overt gangrene can lead to a lower leg amputation. Okay, so let's look how we have used therapeutic angiogenesis to treat PAD. I'm going to talk about some animal studies and then describe the clinical trial, which we'll be starting. Okay, this is a angiogram. So that's basically the same thing they do in the heart. They fill the leg vessels with arteries with a dye. You can see on the left there, normal. At the top is the thigh region, the femoral artery, and then we get to the knee, and then below the knee, you can see those three branching arteries. Look at the right there. Look where the yellow arrows are pointing to. This is a person with PAD. You can see above the knee, the femoral artery is blocked there on the right-hand side with that yellow arrow. Below that, below the knee, look at those vessels completely blocked. And so this person would develop severe pain in the lower leg upon walking. So we're going to be injecting FGF1 directly into the leg muscles to try to grow new blood vessels in a person with this disease. But first, let me show you some work that was done in an animal model. This is the gold standard. It's done in a rabbit. So in the hind leg of the rabbit, you can actually tie off the femoral artery, and this rabbit will develop PAD, and it really closely mimics the human disease. Okay, so this is the rabbit hind limb ischemia model. Let's start at the top there, the aorta, go down to the iliac, and there you can see the femoral artery on the right, we've removed it. So basically remove on the right-hand side of that figure, the femoral artery, and this rabbit develops acute ischemia, chronic ischemia, excuse me, in that hind limb. So actually we have a nice control. On the left there, we have a normal rabbit limb, and on the right is the ischemic limb, and that's the one we're going to be injecting FGF1, as you can see on the bottom there. Day 0 to 20, we do 6 FGF injections, and then we measure blood flow, basically blood flow and new vessel growth in the hind limb of that rabbit. So let me just show you some of that data. Kind of looking inside the rabbit limb there, looking for new blood vessels. And at the top is controls, baseline control, and then a vehicle control. And you can see below 2 doses of FGF1, which were tested, we have 1 microgram per kilogram of rabbit weight, and then 10 micrograms. You can see on the bottom 2 circles there, especially on the bottom right-hand circle, new blood vessels have formed after we injected that leg with FGF1. We can actually look in the muscles of the leg. Here's 2 examples, the adductor muscle on top, gastronemicus on the bottom. Going from left to right, we have placebo, then the low dose and the high dose. And I think on the right-hand side of that slide, you can see all those little new blood vessels which we formed with FGF1. And that's going to lead to improved blood flow in that rabbit leg, okay? So we can actually put little blood flow sensors inside the rabbit and measure blood flow. And on the right there on that graph, you can see dramatically, especially with the high and mid dose, increased blood flow in the rabbit's legs. And those rabbits that have been treated are much happier rabbits. They're jumping around their cages versus the untreated ones really still have severe disease in their hind limb. So with these results, we're ready to take this into the clinic. We're preparing applications to the US FDA, to the Mexican counterpart, which is called COFEPRIS. We like Mexico. They've actually approved Parkinson's disease trial for us there, and also ALS. So they give us really good turnaround. So we've kind of developed a strategy to submit things both to the US government and also to the Mexican. We plan to start that in the 3rd quarter of next year. Let me just show you a brief synopsis. We'll be studying 3 ascending doses of the drug. So we'll do the low dose, make sure it's safe, and then we'll ascend to the middle and high dose, pretty much like we did the heart trial. The first trial, there'll be no placebo group. All patients will get injections once a week for 4 weeks. So we'll get it right into the legs. I'll show you that on the next slide, but it'll be a series of from 6 to 12 injections, and we'll do two types of injections. One will be where the vascular clot is, where the occlusion or obstruction is. And then we'll get down to the lower leg and inject where there's ischemic regions in the calf muscle. And it's a really simple trial to administer because the gold standard test is what's called a 6-minute walk test. So in a corridor or somewhere, a physician will lay out a kind of a walking route, and they measure how far the patient can walk in 6 minutes. So we'll be doing that testing at before treatment and then at 2, 4, 6, and 8 weeks after treatment. So here are those areas of injections I just wanted to mention in the red boxes there. We'll do 2 injections where there's vascular occlusions. That's where the actual clots are. And then we'll do a series of spiral injections through the ischemic calf muscle. And again, we'll do that once a week over a 4 to 6 week period. I just want to end on telling you how we're making our FGF1. I mentioned we originally purified it from cow brain. Obviously, we can't use cow FGF1 in people. And we can't really even purify FGF1 from human sources. It's just in very trace quantities in our body. So we use genetic engineering, molecular biology to grow up FGF1, human FGF1 in bacteria. And you can see here a manufacturing plant. Those silver containers are fermenters. They contain bacteria where we've engineered the human FGF1 gene. So the bacteria actually make FGF1 as part of their own proteins, and we forced it to be made as the most predominant protein in the bacteria. And this just shows a protein gel. I've run thousands of these in my lifetime. This separates, you can see in lane 2 there, that's the entire protein family that's in a bacteria. And going from the top there, the larger proteins down to the bottom are smaller proteins. And there's FGF1 there, right next to the number 19 there in lane 2. It is a predominant protein made in this bacteria. But we need to get rid of all those bacterial proteins because they're toxic, they can cause anaphylaxis. So over a period of weeks, you can see from lane 2 through lane 10, we gradually, with different purification techniques, get rid of more and more of those bacterial proteins to where at lane 10 there, we have pharmaceutical grade, commercial grade FGF1, which is ready for the clinic. Okay, so I'm happy to answer any questions now. And after the Q&A session, Dan Montano will give some concluding remarks. So thank you for your attention.
Thank you, Dr. Jacobs. We have had many questions come in during the presentation. Unfortunately, we only have time to answer a few of them. So to all of our attendees, if your question was not answered, please email to dan@zhityamedicine.com and we will answer it as soon as we can. So the first question is, can the drug migrate to other tissues after being injected to the leg? And what would happen if the drug migrates to another place?
Okay, that's a very good question. You know, we know when we inject our drug into muscle, either heart muscle or leg muscle, it actually stays there for a period of days, 4 to 5 days. Some does escape into the bloodstream, but it gets removed within 4 to 5 hours. So it's really a rapid removal of FGF1 once it gets into the bloodstream. We've done a lot of testing in animals, rats, dogs. In our heart trial, we look carefully and we see no activity of FGF1 other than where we've been injected it into the heart muscle or into leg muscle. So I think the effect is very localized, and we've seen no adverse effects with our drug in all the patients we've treated so far in the clinical trials.
Okay, for the next question, would this drug trigger skin cancer in the area where it has been injected?
Another good question. No, we've looked carefully at any type of initiation of cancer. I can state categorically, FGF1 does not cause or initiate cancer in extensive animal studies. And we've never seen anything like that in our human trials. The FDA has asked us to monitor tumor markers, and they stay completely stable throughout the course of the clinical trial.
Thank you. For the next question, how big is the PAD market?
Well, it's in the US, we have 8 million patients. That's a pretty good sized market. And worldwide, I mentioned, over 200 million people have this disease. Obviously, not everyone could afford the treatment, but I think the market is plenty huge for us.
Thank you, Dr. Jacobs. For this next question, will be our last question to Dr. Jacobs. How many injections does one need to have to regrow the blood vessels in the leg, and how often should this procedure be repeated?
Okay, that's a very good question. So in the animals, we had 6 separate sets of injections were enough to grow the vessels, and they were durable vessels. They lasted for as long as we look for them. So we know in the heart, when we grow blood vessels, they have lasted for 3 years or longer. So we hope this one series of injections in the humans, remember it's once a week for 4 to 6 weeks, will be enough to grow blood vessels in their legs that will last for years and years. So we hope it's adorable. We think it'll be a durable and long-lasting treatment.
Hello, ladies and gentlemen. First of all, I would like to let you know that in case you haven't figured it out yet, we're really new to this Zoom webinar process. I believe this is our 4th one or 5th one. I think the first 2 went better than the last 3. We're learning things and trying new things. So thank you for being patient with us. Our expertise is in pharmaceuticals and biotech, not in making Zoom presentations. First, Zija is a Las Vegas-based biotech company, and we're developing a biological drug that stimulates therapeutic angiogenesis. And we believe our drug can treat the leading causes of death in the world. I would also like to answer one of the questions that was asked of Jack about how often will people need to be treated. Most cardiovascular diseases are ongoing events. Arteriosclerosis and all the things that can cause blockage are a constant battle. When we treated people in the heart, some people did not need additional treatment for 15 years. Some people needed treatment again after 7 years because they continued to smoke, they continued to eat fatty foods, and they did not exercise. I believe that Jack's correct. In the legs, we're going to treat millions of people. But as we treat them and grow new blood vessels, I think over time they may have blockages in other areas. The legs, ones that we fix, may become clogged as time goes by. Since my goal is to be 130 years old, I believe that there may be a lot of repeat treatments over 5, 10, 15 years to continue to regrow blood vessels and to continue to treat the heart and other areas. Our business is discovery. How does the drug work? Where is it safe? How is it safe? How much can we give? How long can we give it? These are things that we are discovering literally every day, every week. The clinical trials that we'll be undertaking in the United States, Mexico, Estonia, and other places in the brain are going to be a lot of discovery. Part of the other thing we do is design and carry out clinical trials to demonstrate that our drug is safe and effective in humans where lack of blood flow is the disease. And then we need to interact with government regulatory authorities so we can obtain approval to sell our drugs to the medical community. Now, this is a booklet, which if you don't have, you can simply email me at dan@zhittamedicine.com and we'll send you this. This is a booklet, which is a quick summary of how our drug works, what's the biology behind it, and some information on the heart treatment, the diabetic foot ulcers, and the Parkinson's. Additionally, if you want a copy of Is Therapeutic Angiogenesis a Potential New Treatment for Critical Leg Ischemia, you can email me and I can either email you back one or I can send you a couple of 100 hard copies. If any of you have any need for a couple of 100 of these or a couple thousand, let me know. Our next upcoming webinar is going to be next Tuesday at 8 a.m. Pacific. It's Alzheimer's disease. Therapeutic angiogenesis is a potential treatment reversing this disease. Over the last 20 years, there has not been a new treatment for Alzheimer's. I would also make a comment that when Biogen was working on a supposed treatment and they thought they had some evidence, their stock went up in one day $61 billion. So great is the medical need for something to treat Alzheimer's that people believe $61 billion in enhanced value in one day. And for those of you that know me, it would make me happy one day to increase my value by $61 billion. The clinical trials that have been addressing Alzheimer's have been looking at beta amyloid plaque. And basically nowadays they have all failed, and that is a failed topic. From McGill University in Canada, they found that the precursor that precedes Alzheimer's appears to be disrupted, decreased blood flow in certain areas of the brain. I think you're going to find Jack's presentation very interesting. I think for your own sake, because all of us fear losing our memory, you'll find this presentation next Tuesday very, very interesting. After that, we have a presentation on the 15th of September. That is going to be at 11 a.m. Pacific time, 2 p.m. Eastern, 8 p.m. Germany, 9 p.m. Saudi Arabia. Multiple sclerosis, is therapeutic angiogenesis a treatment or even a possible cure? The presenter will be Vika Montano, my wife. It's estimated that 2.5 million people in the world suffer from multiple sclerosis. Roughly 3 out of 4 are women. And it's primarily in the northern hemisphere. Now, the reason I think this is very important is because Vika is going to focus a great deal on something called epithelia dysfunction. Epithelia are the cells that make the blood vessels, the live cells that form these tubes that move the blood. If the epithelial cells are dysfunctioning, you're going to have inflammation, you're going to have blockage, you're going to have leakage, you're going to have all kinds of problems. As Dr. Jacobs and Vika started researching multiple sclerosis and found out that these cells, the ones that make up the lining, were having dysfunction, and that led to what many people now believe causes multiple sclerosis. I have absolutely no evidence, but I believe that's also what's happening in the brain and why our brain shrinks as we get older. That is, those epithelial cells down the line, as they get less nutriments, less oxygen, as they function depreciates, they end up in a situation that they die and the vessels shrink, and therefore the brain shrinks. I think this is a very important fundamental concept that's well worth watching. What is the reason we're doing this? Zhittya has an outreach program to make people aware of our medical science. The idea that you can grow blood vessels, believe it or not, is still shocking to most people, even though they've done it thousands of times themselves. And we're trying to make people aware of our science and develop a network of motivated and informed individuals. Over the last 30 days since we started this, we have touched out to over 1 million people. Now, we've only gotten about 10,000 that have joined up with us, but that's still pretty good for 1 month. And some of those 10,000 include leading neurologists, some world leading diagnostic imaging people, doctors from countries all over the world that are interested in us succeeding, and people who are interested. We presently have over 40,000 followers, probably closer to 50,000 today. And over the next 60 days, we will probably be reaching out to another 1 million people, because we believe that the world has to know what we're doing, so that when we have the science, the support, and the evidence, people will listen. If you have any questions, please free to email me, dan@zhittямedicine.com. Visit our website. If you want any white papers on anything we're doing, we're here to inform the world. Now, I make this comment because we'll be doing webinars, we're having other outreach campaigns. We believe that we have something, and I hope and pray that you'll be there to join with us. Saying goodbye, and thank you very much for your time.